Good morning, everyone. My name is Marie Lauigan, and I am a technical support scientist at Advanced Cell Diagnostics. Before we start, I do have a few points to go over. The audience will be muted during the presentation. Please feel free to use the chat feature to ask any questions while the presentation is in progress, and we will do our best to answer them. In the event that we have missed your question, we will follow up with you offline after the webinar. Now, I'd like to begin on how to visualize your research breakthroughs with RNAscope and the BaseScope assays. The topics that we'll be covering today include an overview and an introduction to RNAscope and the technology. We'll go over the RNAscope assay workflow. We'll discuss performing the RNAscope manual and automation assays. And we'll spend some time on troubleshooting tips for the RNAscope manual assays and automation assays. I'll also provide a brief introduction on getting started with the BaseScope manual assay, and at the end we'll address questions and answers. Here's the RNAscope workflow. We are working to permeabilize the cells or the tissue in order to target and hybridize our target RNA. We will amplify the signal, and you'll be able to visualize with morphology and quantify with a single-cell expression. With RNAscope, it utilizes a unique probe design that allows for simultaneous signal amplification and background suppression to achieve single molecule detection seen as a single punctate dot. This can be done with virtually any genome, any gene, or any tissue. Here is a brief overview of the probe design. With a customer gene of interest, you can provide the accession number for your gene or the target sequence to our probe bioinformatics team. And using the ACD alignment algorithm, they can identify low homology and make the standard probe configuration, which consists of 20 Z oligo pairs equaling to about 1,000 nucleotides. With the RNAscope technology and the double Z probe design, the lower region of the Z is a 8 to 25 base sequence. There's a spacer, and then an upper region of a 14 base tail sequence. With the double Z probe design, the target-specific binding site is about a 50 base region on the bottom targeting to your transcript RNA. And on the top of the double Z is a preamplifier binding site that serves as a foundation for building the amplification tree. The standard 20 double Z oligo design can target intact RNA as well as fragmented regions of RNA. The RNA target region covers about 1 kb or 20 pairs of about 50 bases. The mRNA transcript detection is highly specific and provides robust signal amplification. And so how does RNAscope work? Here is a schematic cartoon showing the signal amplification. Here you see below the target RNA transcript. This is the double Z target probes binding to your target RNA. The preamplifier then binds to the top of the double Z probe, followed by subsequent binding of the amplifiers to your preamplifier, and the label probes binding to the amplifier. This is then done across in succession, again with 20 Z pairs, 20 double Z pairs. For nonspecific targets, with a double Z probe design, this makes nonspecific amplification not possible, as you cannot have amplification occur on only one Z. And so I'd like to discuss the RNAscope assays that we currently have available. Here is a chart of all the RNAscope assays. We have the brown chromogenic assay available in the manual, VENTANA or LEICA platforms. We also have the chromogenic red assay available in the manual, VENTANA or LEICA platforms. We have the RNAscope DUPLEX assay that is available in the manual and the LEICA bond. And we also have the MULTIPLEX fluorescent assay available in the manual and the LEICA bond as well. Highlighted in red are the chemistries and the dyes used in order to be able to visualize your signal. And highlighted in green below are the probes that are required in order for you to run the assay. Here is a cartoon for the single-plex amplification for the red assay or the brown assay. Again, you'll see the double Z pair of the preamplifier binding to the double Z of your probe, the amplifier then binding to the preamplifier, and the label probe binding to the amplifier. For the red assay, it utilizes the alkaline phosphatase enzyme and is then visualized with the fast red substrate. For the brown assay, similarly with the amplification tree, it does use a horseradish peroxidase enzyme, and the substrate is the DAB to get the brown dots. For the duplex assay, you have two independent trees that are built for each channel. For the C2 channel, we have the alkaline phosphatase enzyme utilizing the fast red chromogen. And for the C1 tree, we have the horseradish peroxidase using the green chromogen. Do note that you can have the HD duplex run on the automated platform on the LEICA Bond RX, and it can utilize either a red-brown chromogen staining or red-green chromogen staining. Do note that if you're running the duplex assay, the C1 probes are normally kept at 1x concentration, while the C2 probes are provided as 50x. To make a duplex probe mixture, you'll need to mix the C2 probes in a 1:50 dilution with the C1 probes. It is possible to view the C2 probes only if you use the blank probe C1 as a diluent and mix it at a 1:50 dilution. Here we have the scheme for the multiplex fluorescent assay. Again, similar of having the double Z bind to your target RNA, the preamplifier and amplifier with the label probe. Here it utilizes fluorescent labeled probes in the form of the Alexa 488, the ATO 550, or the ATO 647. Similar to that of the duplex probe, the C1 probes again are provided in 1x concentration, while the C2 and C3 probes are at 50x. And again, to make the 3-plex probe mixture, you will need to mix C2 and C3 probes at a 1:50 dilution with the C1 probe. Also note that the multiplex fluorescent assay was recently released on the automated platform to be performed on the LEICA Bond RX. And now looking at the RNAscope single-plex manual assay workflow. The single-plex chromogenic assays are done and completed in 4 major steps, with a total time to completion of about 7 hours. This occurs with the pre-treatment of your tissue, your target probe hybridization, the building of your amplification tree, and the staining and detection. Do note that this protocol is the workflow for FFPE samples, and we encourage you to visit the website to make sure you are getting the correct user manual for the correct sample type, as this may differ. Similarly, this is the workflow for the fluorescent assay, again in which you would have the 4 major steps of pretreatment, hybridization, amplification, staining, and detection. For completion, the fluorescent assay is about 5.5 hours. And do note that this protocol is for fresh frozen samples, and again, pretreatment conditions will vary based on the sample type. So again, do make sure that you visit the website to download the correct manual or technical note for the sample type you are working with. Most of the assays can be split into 2 days, but we do recommend that you refer to the user manual for the recommended stopping point. If you were to break up the assay in 2 days, you would complete sample preparation and pretreatment in 1 day, stop, and complete the RNAscope detection assay on the 2nd day. Again, please refer to the user manual for the detailed instructions as well as where the optional stopping points would be located. And so some tips for running the RNAscope manual assays. Here we have some basic tips to follow. We do highly recommend that you follow the protocols exactly as described in the user manuals. The user manuals are quite lengthy, but they are a wealth of information full of how to best run the assay to achieve optimal results. We do advise that you review the sample pretreatment conditions, as pretreatment can differ based on your sample type and how your sample is prepared. We highly recommend the use of control probes to ensure that the technique is working with the RNAscope assay, as well as to provide an RNA quality check in your sample. We also recommend that you review that you're using all the required materials via a checklist that can again be found on the website. Again, the checklist does differ depending on the assay that you are running, so do ensure that you have the checklist for the correct assay. A brief overview of what the checklist might include would include the image hydrophobic barrier pen. We have heard of other PAP pens being used, and so we highly recommend the use of the image hydrophobic barrier pen to keep the hydrophobic barrier intact on your slides. We also highly recommend the use of positive and negative controls, as again, this serves as an assay workflow technique check and to confirm the RNA quality in your samples before proceeding to your target probes. We recommend the use of a hot plate or steamer for the pretreatment of your target retrieval step, as this is what we have worked with in-house and validated. And we also highly recommend the use of SuperFrost Plus slides, as with the use of any other slides, we have seen severe tissue detachment. Again, based on the assay that you are running, there are different checklists, and so please review them accordingly. Here we have an image of either the hot plate for doing your target retrieval or the steamer, which we both have protocols for. When using the hot plate or the steamer, it's most important to pay attention to time as well as the boiling temperature, of which you should keep it between 98 to 102 degrees Celsius. Again, the time will differ depending on what tissue you're working with, so please do refer to your user manual. And here's what you would see with the reagent kit content for manual assays. This should include the pretreatment reagents, the RNAscope detection kit, as well as the wash buffer. Do note that you may need to either warm your probes at 40 degrees for 10 minutes before use, or warm up the 50x wash buffer at 40 degrees for 20 minutes if you do notice precipitation. Here we have the high-VZ hybridization oven in which you would complete the protease digestion and the hybridization steps. The high-VZ oven is required as it provides both the temperature and humidity control that's necessary to obtain optimal RNAscope results. Here we also have accessories for completing the washing steps in between each amplification step. To the left is a tissue tech washing tray, which you can find by any major lab supplier. And to the right is the Easy Batch slide processing tray, which is easy and convenient for loading multiple slides and for washing and doing the hybridization steps. If you are interested in the Easy Batch slide processing tray, this is something that ACD can provide, and you may contact your account executive if you are interested. It's important to follow the workflow guidelines when running the manual assays. And so here below is a quick list of the guidelines that you should be following, such as applying all the amplification steps in the correct order. Ensure that you're not letting the slides dry out, as this can affect the outcome of the assay. Do make sure that you're not altering the protocol in any way, and that you're following the user manual as is, and that you're reading the user manual per the assay that you are running. So some brief tips for the RNAscope manual assays, in particular with the RED assay. Here we see the detection of simian immunodeficiency virus, or SIV, with the RNAscope HD RED chromogenic kit. It is very important that you use EcoMount as a mounting medium, and that you avoid any dehydration of the sample with alcohol, which can cause a diffusion of the signal. Rather than dehydrating with alcohol, the sample should be dried in a 60-degree oven for 15 minutes before mounting with EcoMount. The samples can be left in longer than 15 minutes to ensure that there is no moisture retained on the slide prior to mounting. Some tips for the RNAscope DUPLEX manual assay. This is the workflow for the 2.5 HD duplex assay. To the left, you'll see the pretreatment all the way down to the probe hybridization. You'll also notice that you develop the C2 probe first with a fast red chromogen, followed by the development of the C1 probe with the green chromogen. The three arrows to the left show optional stopping points in the assay so that you can, again, complete this as a 2-day assay. But this highlights optional stop number 4, which is the best stopping point for the duplex assay. After probe hybridization, you would then stop in 5x SSC, leaving the slides in room temp overnight. This can be made by diluting any 20x SSC provided by any major lab supplier. The dotted line will then show everything to the left would be completed on day 1, while everything to the right of the dotted line would be completed on day 2, starting from the duplex AMP1 and completing all the way down to drying and mounting with VENTANA. When getting started with the RNAscope HD duplex assay, we do have an intro pack available. Here you'll see the list for the human-specific tissue intro pack, in which we include the Abamax HeLa control slide. We will include the duplex positive control probe for human, as well as the duplex negative control. It will also include the reagent kit for the duplex assay, the wash buffer, the hydrophobic barrier pen, and the VENTANA mounting medium. Please do review the user manual and the quick guide to familiarize yourself with the workflow prior to starting the experiment, and ensure that you have the checklist for this assay to confirm that you have all of the required reagents before starting. The probe configuration for the duplex assay is as below, where the C1 probe is visualized in green using the HRP in the green substrate, and the C2 probe is visualized in red, again using the alkaline phosphatase and fast red substrate. Below is the color combinations for the control probes, with the positive control having PPIB, a high expressing marker in green, and polar 2A, a medium expressing marker in red. The negative control utilizes the bacterial gene DAPB in both channels. We do recommend that you place the lower expressor in the red channel. Again, please feel free to visit the website for any tips and frequently asked questions for the manual assay. Here we have a picture of the duplex assay with the CD248 marker in green and the PDG-FRB marker in red in human colon cancer FFPE tissue. Again, you want to make the duplex probes by mixing the C2 probes 1 to 50 with the C1 probes. Again, if you want to view the C2 probes separately, you would use the blank probe or probe diluent instead of the C1 to do your 1 to 50 dilution. We also require that you use VENTANA as the mounting medium to help preserve the green and red signal. Similar to the red, we also ask that you avoid dehydration of the sample with alcohol, again to avoid a diffusion of the signal, and that the sample should be dried in a 60-degree oven for at least 15 minutes before mounting. Again, the slides can be left longer to ensure that there is no moisture retained on your slide. Some tips for the manual assay on the multiplex fluorescent. Here we have the image of the multiplex assay. With the multiplex assay, there are 3 color module options available to provide for 3 options of color combinations. For example, AMP4 Alt-A would show the C1 probe in the green, or the Alexa 488 dye. It would show the C2 probe in orange, or the Atto 550. And it would show the C3 probe in far red, or the Atto 647. Please note you only need to choose 1 color module when running the assay. And again, similar to that of the duplex, the C1 probes are 1x concentration, while the C2 and C3 probes are 50x. And so we do recommend that you mix the C2 and C3 probes with a 1 to 50 dilution with the C1 probe or with the blank probe. We highly recommend the use of the Prolonged Gold as a recommended mounting medium to help preserve the fluorescent signal. Again, this shows the 3 independent amplification trees that are built for each channel, which will allow you to visualize multiple targets in one sample, so that your C1 target is a tree that is being fluoresced by the Alexa 488. Your C2 probe has its independent tree, then being fluoresced by the ATO 550, and you have the C3 marker being fluoresced at the ATO 647. Moving on, we do have available the RNAscope on automation platforms. We have it with the VENTANA Discovery Ultra and the XT. Now this allows for a fully automated RNAscope assay on both the Discovery Ultra and the XT platforms. For the RNAscope 2.5 VS reagents, they are available in the red singleplex or the brown singleplex. The reagents provided by us include the target retrieval, protease, DWAX, your custom target probe, and the amplification components. This then utilizes the detection substrate from VENTANA, as well as some VENTANA and Roche accessories in which we note out in the user manual to purchase before use. If you are interested in running the 2.5 VS assay, we do ask you to contact ACD support to schedule the RNAscope VS 2.5 training as well as the software update for your machine. Tips for the automation assays, specifically on the VENTANA systems, is that you check the instrument maintenance, that you're optimizing the software settings if needed to troubleshoot reagents, and that you review the sample pretreatment recommendations. In particular, for checking the instrument maintenance, it's important that you perform decontamination protocols every 3 months to prevent microbial growth on the equipment, and that you're using the appropriate buffers for the RNAscope assay. In regards to optimizing the software settings, for the Ultra only, we recommend that you uncheck the slide cleaning option and that you're using the appropriate hybridization temperatures, as this may differ between the XT and the Ultra models. Again, please refer to the user manual online. We do have available for either the XT platform or the Ultra. Here's an example of the staining with 2.5 VS brown assay on the VENTANA Discovery Ultra. And the top row is the DAPB negative control, and the bottom row is with the human PPIB positive control probe on 4 different tissue samples with breast, lung, lymph node, and skin. You'll see a clean DAPB negative control, no background, and high robust staining with a human PPIB control probe. Also available for the automation assays is the LEICA Bond RX. Here we'll talk about the singleplex assays in which you can run either a singleplex red or brown assay, fully automated on the instrument. Again, LEICA provides the target retrieval as well as the refined kits for detection substrate, and ACD will provide the protease, wash buffer, your custom target probe, and the amplification components. Again, just with the VENTANA, if you're interested in running the 2.5 LEICA assay for the first time, we suggest that you contact ACD support to schedule an RNAscope LS 2.5 training as well as a software update. With the LEICA Bond RX, the assay workflow is seen below, the total runtime being about 10 hours for the singleplex assay, in which you would be preparing your materials, setting up your protocols, the time on the LEICA Bond RX, which includes the baking, pretreatment, target probe detection, and the offline steps, including the counterstain, dehydrating, and mounting. Again, here are samples of staining of the 2.5 LS brown assay on the LEICA Bond RX, the top row being the DAPB negative control probe, the bottom row being human PPIB positive control probe, and the 4 different tissue sample types. We also have the automation assays with the DUPLEX chromogenic assay. And here's the assay workflow on the LEICA Bond RX. Fully automated, the assay is roughly 14 hours. This is for doing a DAB and red detection, brown and red detection using the DAB and red refined kits from LEICA. Here you see the assay workflow, which does look similar to the chromogenic manual duplex assay, in which you would do the pre-staining, pre-treatment up to probe hybridization. You would then develop your C2 channel first, then with the LEICA red refined kit, and then you would move to the C1 channel and then visualize with the LEICA DAB refined kit. Please note that the duplex assay is also available to do red and green chromogen staining with the LEICA duplex, the green chromogen replacing the LEICA DAB. The green chromogen would need to be done offline, and the green chromogen is also available through ACD. Here's an example of staining of the LEICA duplex assay with the red and brown chromogenic staining. You'll see the GRIN2C marker in dark brown in the middle of the tissue and the PVALB marker on the periphery in mouse brain tissue. Recently released, we also have the LEICA Bond RX assay for multiplex fluorescent. You'll see in this workflow that you're developing each channel sequentially with a blocking step after each fluorophore. This also introduces TSA as part of the amplification system that would need to be purchased from another vendor, PerkinElmer. Please note that you may also choose to complete IHC afterwards, in which you can do the bond wash step in place of DAPI. For further information on completing RNAscope ISH and IHC, we do have some information on our site, and you can also feel free to contact technical support. Here's the information on what you would need to purchase from PerkinElmer. We have validated the assay using the TSA Plus system, and that uses the TSA Plus fluorescein, cyanine 3, and the cyanine 5 system. We've also found that our assay is compatible with the Opal 7-color F IHC kit, as seen below. Here's an example of some neuroscience markers that were stained using the LEICA multiplex fluorescent kit. You have the VGLUT2 marker in green, you have the VGLUT1 marker seen in orange, and you have the VGAT marker in white. And this was done on mouse fresh frozen brain. As previously mentioned, it is possible to do ISH and IHC application together. Here we see the OLFM4 marker using the RNAscope chromogenic brown assay and the lysozyme antibody with IHC. Again, you may visit the site below for more tips and information regarding dual ISH and IHC. Similarly, you can also perform it with the multiplex fluorescent kit as well as antibody staining seen here. And now I'd like to spend some time on getting started in troubleshooting the RNAscope assay. So the most important thing when starting the assay is how to get quality data, and that includes qualifying your samples with controls. Seen here for image analysis is a semi-quantitative scoring guideline that we use for RNAscope, a scale from 0 to 4, 0 being no staining or less than 1 dot per cell, 1 with 1 to 3 dots per cell, a score of 2, which is 4 to 9 dots per cell, a score of 3, which is 10 to 15 dots per cell, but less than 10% of the dots are in clusters, or a score of 4 in which there are greater than 15 dots per cell and greater than 10% of the dots in clusters. For qualifying your samples using the controls, we'll start to the left with the control slides, for example, with a HeLa control slide that we can provide, in which you would run the negative control, DAP-B, and the positive control, PPIB, to confirm as a technique check. You'll see in the next image the QC check that the PPIB has a staining greater than 2, with a DAP-B staining less than 1. If your QC yields these results, you may then proceed to testing the control probes on your sample for an RNA quality check. That would include running the negative control probe, DAP-B, on your sample, as well as the positive control probe, for example, PPIB. The next image you see is the staining of the DAPI and the PPIB, again, passing QC check. If you have passed the QC with the sample and RNA quality check, then you can suggest that you can run your target probes. If at this point it fails, we would need to ensure that you can verify the technique, potentially check the RNA quality with new samples, or you may need to perform assay optimization. Keep in mind that you should always be starting with the recommended standard conditions when starting the assay, and you can always refer to the troubleshooting guide. And so for optimizing your assay, you might wonder why you need to optimize your assay and what would cause that. Here are a couple of things that could require for optimization of the assay, including underfixing your tissue, perhaps overfixing your tissue. You may be working with special sample types such as cell pellets or even special tissues as lymphoid tissues. These may require certain milder or more extended conditions when running the assay. Below are some factors that affect the RNAscope assay performance as well as the solutions. One of the top factors being fixation conditions not being optimal. The quick solution is that you should fix samples as recommended per our guidelines. For example, with FFPE tissues, you should use 10% neutral buffered formalin fixed for 16 to 32 hours at room temperature. When optimizing your assay and your sample, this can be done in 3 easy steps. Again, we always recommend that you start with the standard conditions. From the standard conditions, and you can observe the staining pattern, whether the high background may lead to looking over-digested, most likely the tissue is underfixed. But if you're seeing no signal or weak signal and the tissue is looking under-digested, then the tissue is probably overfixed. Seen in step 2 and step 3 are the target retrieval time to the left and the protease time to the right. Both of these can be moved on a sliding scale depending on where your tissue sits based on fixation. If your tissue is underfixed or perhaps you have special sample types, then you can decrease your target retrieval time or decrease your protease time. On the other side, if your tissues are overfixed, you can either increase your boiling time or increase the protease time. Do note for cultured cells, the protease is diluted 1 to 15 in a 1x PBS, and for fresh frozen samples, only protease pretreatment is required. It's performed at room temperature and not at 40 degrees. Again, for the specifics of protocol for the sample type, please feel free to visit the website. Similarly, on the automated platforms with the LEICA BondRx and the VENTANA XT or Ultra, you can adjust your ER2 time in increments of 5 minutes in protease for the LEICA BondRx. You can also reduce temperature. And for the VENTANA, you can either increase or decrease your pretreat and or CC time. Again, based on the user manuals for the assay workflow and the pretreatment optimization guidelines, please feel free to refer to that. So you've gone through qualifying your samples, you've optimized the assay, and this is what an example of a successful RNAscope assay should look like with the CHROMOGENIC HD Red assay. To the left, you'll see a clean DAPI negative control probe. In the middle, you'll see robust staining with a positive control PPIB probe. And with your target probe, you'll see very specific staining in your tissue. To further get quality data with the RNAscope, you may have to perform some troubleshooting experiments. We'll start by looking at troubleshooting tips for the RNAscope multiplex fluorescent assay. In this slide, we're looking at sample digestion as being the issue. In the top panel, we have fresh frozen mouse kidney. In the far left picture, we have the protease for treatment being done at 40 degrees, in which you're seeing overdigestion and a loss of signal. We've then optimized so that you would complete the protease for incubation at room temperature, and you see now that the overdigestion has been solved, and that you also can see the red and the green channel signals, which you could not previously see in the overdigested sample. Similarly, in the bottom panel is fresh frozen mouse brain, in which, again, you're completing the protease 4 at 40 degrees with overdigestion and loss of signal. And then to the right, once you complete the protease 4 at room temperature, you can see the signal clarity of your staining. So therefore, it is important that the pretreatment temperature can have a great effect on the success of your assay. And the solution would be to perform the pretreatment according to what we specify in the guidelines. Also taking a look at sample digestion is sample thickness. So sample thickness we do recommend for fresh frozen tissues between 10 to 20 microns. You can see across the images that as you increase in the sample thickness, that the intensity of the staining also increases. Normally we do recommend, and where most customers sit, is between a thickness of 12 to 15 microns. For this experimental condition, the tissues are fixed at the standard conditions with 10% NBF for 15 minutes, then doing a protease 4 incubation at room temperature. One of the main issues that we see with the multiplex fluorescent assay is autofluorescence. With FFPE samples, they do have an inherent autofluorescence, and so this may be difficult to determine where is true signal versus the autofluorescence. The quickest solution is that you use the appropriate background correction software to help reduce the autofluorescence, as this would help to reduce the non-specific staining and help to increase the threshold of your signal. Looking at some of the multiplex problems and solutions, if the source may be microscopy, the issue could be no or weak signal or non-specific signal. It could be due to the wrong filter setting, wrong exposure, or perhaps the inappropriate imaging enhancing with software. And so we would highly recommend that you use the correct filter settings, that you're not auto-exposing your sample, and that you can at least verify signal with the naked eye first, and if possible, use a known image enhancing software. If the source of the issue may be coming from your sample, it could be due to a compromised RNA quality, sample preparation, which can lead to high autofluorescence background on the sample. And for the solution, we'd suggest you try a new sample with a good RNA quality, that you follow the pretreatment guideline, and that it's very important that you perform the assay with the 3-plex positive control and the 3-plex negative control to always assess the RNA quality. Again, you should always check your signal with the naked eye under the objective lens first. And so after all of the optimization, here is what we would see for an example of a successful RNAscope multiplex fluorescent assay. This is utilizing the AM4Alt-A, and so the C1 probe, Polar 2A, is visualized in green. The C2 probe, seen here in red with the human PPID. And the C3 probe here in white, which is the human UBC, and the final image of 3 probes merged together. Again, you can use different AM4Alt reagents depending on what color you'd like to visualize your marker in. And so some troubleshooting tips for the chromogenic assay. First, let's take a look at overdigested samples. Here we have FFPE tissue that was fixed for 12 hours on the left-hand side. And with the standard 15-minute target retrieval boil, we see that we've destroyed the morphology, the presence of ghost nuclei, high nuclear background, and very weak hematoxylin staining. Because it was underfixed, we overtreated the tissue, and so now what we can do to optimize it is modify the boiling time from 15 minutes to 8 minutes. Then you can see on the right-hand side that we now have intact morphology, a strong hematoxylin staining, and we were able to reduce no background in the tissue. Again, this is seen in the DAPI negative control. Similarly, this is the same tissue except with PPID staining. You can now see the difference in that there is a stronger hematoxylin staining, and there's actually better signal resolution once we've modified the target retrieval boiling time. Next, we'll take a look at underdigested samples. Here we have FFP tissue that was fixed for 72 hours, which is almost double than what we would recommend. Here you see that there's low signal intensity as well as dense compact nuclei, and so the 15-minute boil is actually insufficient for the tissue. Here, this is actually staining with the PPIB probe, and so you can see a significant impact on signal intensity. For the optimization, we then decided to increase the boiling time from 15 minutes to 30 minutes. You do see stronger signal intensity and better signal resolution. Another issue to troubleshoot could be the presence of nuclear background staining. Here we have an assay that was run on the LEICA Bond RX with the LS Brown, DAPI negative control, and you see the presence of nuclear hazy background. This was done at the standard pretreatment conditions of a 15-minute ER2 and 30-minute protease. For optimizing on the LEICA, you can increase the ER2 time in increments of 5 minutes, and the protease can be done in increments of 10 minutes. For this human tonsil FFPE tissue, we decided to increase the ER2 time to 20 minutes and kept the 30 minutes of protease, and now you can see the significant reduction of background. You can also see high background, which may be due to drying or insufficient deparaffinization. If the background type you're seeing is cytoplasmic or nuclear, again, this could be due to drying between the amplification steps. And so we recommend that you always completely cover the tissue when applying the reagents and that you process them one at a time to prevent drying unless you have the easy batch slide system, and that you are using the oven at the appropriate temperature, also while using the hydrophobic barrier pen to ensure that the hydrophobic barrier is kept intact. If the type of background you're seeing is extracellular, the cause, again, could be incomplete paraffin removal or suboptimal tissue preparation. And so we always recommend that you're using fresh reagents for deparaffinization to prevent if there's any contamination, and that you prepare your tissue samples according to the ACD recommended procedures, which, again, are all available on our website. Another issue that we've seen is sample detachment. So here we can see in xenograft tissue of the standard baking protocol and the significant loss of tissue. And what we've done is increase the baking step to 1 hour, and you've seen significant retention of the sample. Where this can most likely be root cause to is the wrong type of slides being used. That's why we highly suggest the use of SuperFrost Plus slides, as that's what we've used and validated internally. This, again, could also be due to suboptimal tissue preparation, which is why it's always important to follow the guidelines. And just as what we did for here is that you can bake your slides for a longer period of time. Another area in which we're seeing tissue detachment is most likely seen in fixed frozen samples. So this slide does have a lot of information on 3 modifications that we would recommend at first, followed by the 4th modification only if the first 3 modifications fail. We do recommend for fixed frozen samples that you are sectioning at a 7 to 15 micron thickness. Again, always use SuperFrost Plus slides, and we have the Fisher Scientific catalog number seen below. If you're going to be baking and drying your slides, do use an active air circulating oven and not the HYBRIDIZATION oven. So again, there are the 3 modifications, and again, feel free to contact ACD technical support for further guidance if you are faced with this issue. Not as often, there is sample detachment with FFPE samples, but if so, we would include these 2 modifications of introducing extra baking steps. Again, the most important part is that you're using the SuperFrost Plus slide and that you're maintaining boiling of target retrieval at 100 degrees, and that, again, you're baking and drying your slides in an active air circulating oven and not the HYBRIDIZATION oven. Here we'll look at a brief issue of insufficient sample fixation. To the left is a tissue that was fixed optimally for 24 hours. To the right, you'll see completely overfixed tissue in which they fixed for 3 weeks. You can see the significant reduction of signal intensity by overfixing your tissue, and to some end, you would then have to optimize to increase your pretreatment conditions to overcome the overfixation of your tissue. Some other issues that might occur is that you could be procuring your samples from either a clinical site or a vendor who doesn't provide you detailed instructions on how the tissue is prepared. And so you would need to do some assay optimization and always start with the standard conditions. Again, from one of the previous slides, starting with standard conditions will then tell you on which way you can optimize. Now I'd like to switch over and provide a brief introduction on the BaseScope assay that was recently released. You can visit below for more further information on the BaseScope assay. The BaseScope assay, it's next-gen in situ hybridization that's ultra-sensitive, and it's allowing for the detection of exon junctions and mutations in cells and tissues with morphological context. To the left is the current schematic for RNAscope 2.5 assays, and to the right is for BaseScope. For RNAscope, it requires 20 ZZ pairs per each mRNA or long non-coding RNA target, whereas the BaseScope assay only requires 1 ZZ pair. The RNAscope assay allows for simultaneous detection of multiple RNA markers, as well as DUPLEX and CHROMOGENIC or 3-plex fluorescent, and can be automated and manual, while the BaseScope assay will only detect for exon junctions, short sequences, or point mutations. And it's also only available as a chromogenic red manual assay. Again, please feel free to visit the website for further information. Here we have the assay workflow for BaseScope, which is roughly 30 minutes longer than the RNAscope assay. To the left, you'll see that the pretreatment conditions are fairly similar to that of the RNAscope assay. Then you would complete the amplification steps from AMP0 to AMP6, followed by the fast red detection. Please note that the BaseScope amplification reagents cannot be interchanged with RNAscope as they are working on different chemistries, and it is different technology. Here is the product insert for BaseScope to show what you would receive if purchasing the BaseScope reagent kit. The reagent kit contains the universal pretreatment so that you can use it for all tissue types. For example, protease 3 would be required for FFPE tissues, while the protease 4 would be used for fresh frozen tissues. And again, just as a reminder that the BaseScope amps are unique to the BaseScope platform and cannot be interchanged with the RNAscope kit. For the control probes of BaseScope, depending on the application of the assay, if you're looking at splice variants, we do have a 1 ZZ length probe, positive and negative control probe, available in human or mouse. And if you're looking at either short sequence or challenging targets, we have a 3 ZZ length probe used to compare for target probes within 2 to 4 ZZs, again with the PPIB and the DAPB, positive control and negative control. Again, here's an example of a successful BaseScope assay. To the left, in which you would see the marker in a short target in the human bladder tissue. In the middle, you would see the 3ZZ human PPIB positive control probe. And to the right is the DAPB 3ZZ negative control probe. That concludes the presentation, and so I'll go over some frequently asked questions as well as address some questions that we have. So the RNAscope assay compatibility with different tissues. The RNAscope manual assay can be used for FFPE, fresh frozen, fixed frozen, or cultured cells. Again, we have all this information on the website through technical support if you go through our technical notes. Currently, the RNAscope automated assays are primarily supported for FFPE tissues. But again, please visit the website for the user manual selection guide as well as any particular protocols and technical notes for specific sample types. A frequently asked question might be, why did you not see signal with positive control? And it could, again, be due likely to RNA quality likely being degraded, or it could be due to sample preparation and pretreatment not being optimal. And so again, we always advise you to refer to the user manual. Always start with standard conditions and then optimize accordingly. Some other questions that have popped up on the automated platforms is, with the VENTANA can you perform a dual IHC-ish assay on the Discovery Ultra and XT platforms? We do have limited information, but if you are interested in being able to do dual-ish and IHC with the Discovery Ultra or XT, you can contact technical support and we'll be able to help provide more and further information. For the LS duplex chromogenic assays, what colors are available for the LS duplex, and will it work with other chromogens provided by other vendors? Currently, we are using LEICA's DAV Refine and Red Refine kit to do the brown and red chromogenic duplex. We also can do red and green chromogenic duplex, with the green substrate being provided by ACD as an accessory item. For the LS multiplex fluorescent assays, you might wonder how flexible is it to switch fluorescent dyes, and it's actually quite flexible in the sense that you are preparing the TSA plus the fluorescent dye, and you may change it accordingly to which fluorophore you'd like to see for which channel of probe. Again, if you have more questions, specific questions, you can contact ACD technical support. Briefly, just to give you information on how to best contact us, you can always contact us via email, support@acdbio.com. You can also contact us by phone in the U.S. at the phone number provided below, and our phone lines are open between 8 a.m. and 6 p.m. Pacific Standard Time, and feel free to visit the support resources available on the website. We do have technical support scientists located in Europe, in China, and in Japan, and so if you have any questions, we can make sure that they are forwarded to the correct technical support scientists, and so you can email us at support@acdbio.com. A couple more questions that have come in. So someone is asking, what are the differences between the manual proteases that you recommend for the various sample types? So the difference in the protease 4 and protease plus is the enzymatic strength and the activity. And to use protease 3 as the standard for reference, protease 4 is approximately double the enzymatic strength of protease 3, while protease plus is approximately half the strength of protease 3. If ranking the different proteases in terms of enzymatic strength, from weakest to strongest, it would be protease plus, protease 3, and protease 4. If you're wondering which protease to use for the tissue type, again, this is all spelled out in the technical notes that we have available online, or if you have specific questions, again, do feel free to contact technical support. Another question that came in is what would you do if you have seen bubbles in your tissue? And we've seen that this phenomenon typically happens in tissues such as in tonsil, and we believe that it's probably caused during the target retrieval step. So one recommendation that we can mitigate for the presence of bubbles is that you can rinse the slides off in distilled water after your target retrieval step. Then you can place the slides in fresh 100% ethanol and leave it for 15 minutes. After that, you may air dry the slides and proceed to the protease step. So you may try this to see if this helps to reduce the bubbles that you're seeing in tissue, which again is mostly caused by the presence of moisture on the tissue. And so that's why we would recommend to increase the time of how long you may leave it in the 100% ethanol. That concludes the webinar for today. In the event that we didn't answer your questions, we will get back to you offline, and so we will get down that information, but please feel free to email us again at support@acdbio.com. Visit the website for, again, any useful information on the user manuals, on contact information, previously recorded webinars, or any other resources that may help you with running the assay. If you have any questions, again, feel free to let us know, and thank you all for attending this webinar today.